Next, we're honored to have Dr. Michael Berger, who's now co-director of the Marie Jose and Henry Kravis Center for Molecular Oncology. He's an attending and molecular diagnostic service in the Department of Pathology at Memorial Sloan Kettering. Appreciate your coming today, and he's going to talk about trends in precision medicine.
Great. Yeah, thank you very much. Good morning, everybody. So I want to, first start with my disclosures. And also thank Tom for giving such a great overview. As you probably appreciate, the testing for these mutations has evolved quite a bit over the years. So when I first started at Sloan Kettering about 12 years ago now, the workflow for a patient with newly diagnosed lung cancer was very complex. By that point, there were already a number of biomarkers that were known, but no sort of single systematic way to test them all. So a patient with lung cancer would have their tumor split across lots of different tests using different technologies to look for different markers. And as the number of markers continued to increase, it created a role for next generation sequencing. So this is the basis for comprehensive cancer gene panels that Tom mentioned. As an example, the MSK Impact panel is what we use at MSK. It was established first in my lab and has become our institutional panel for both clinical and research at MSK, and encompasses 505 genes where we sequence both the tumor and the matched normal for the discussion that we were just having. The clinical workflow is shown here. We have a very large database of the mutations that we've detected, annotated with the OncoKB knowledge base that Tom mentioned I'll say a little bit about. And since this was first established in 2014 for clinical testing, and we got FDA authorization in 2017, we have now sequenced over 86,000 tumors and matched normal DNAs from over 68,000 patients across all cancer types. So this has been the basis for screening patients for clinical trials, for a lot of the research and data mining that we do. But each of these represents a patient who has come for treatment, who's gotten a comprehensive cancer genomic profile. So I'll sort of walk through some of the benefits, both at the individual patient level and the cohort level. Although Tom already did a nice job of summarizing how each individual patient can benefit from this information for therapy selection. So we rely heavily on the OncoKB knowledge base. This was established at MSK, sort of crowdsourced, but internally to capture the biological and clinical significance of individual mutations, not just at the gene level, but as you heard, not all mutations are the same. And the significance of a given mutation may differ from one cancer type to another, or at least the clinical evidence of the significance of a mutation may come primarily from, let's say, breast cancer, whereas the mutation may also occasionally be detected in colon cancer. So the whole tiering system, you can find the OncoKB annotations at this website, and this was granted FDA recognition, the first and I think only somatic knowledge base to get that distinction. So each patient that comes to MSK, now all patients with metastatic cancer and increasingly patients with earlier stage disease would have MSK Impact performed on their tumor DNA and matched normal. The diagnostic report includes lots of different classes of alterations, mutations, amplifications, deletions, rearrangements, signatures like tumor mutation burden and microsatellite instability that can guide decisions regarding target therapy or immunotherapy. And the OncoKB annotations are shown as well. Depending on how you define, we find at least one targetable alteration in between 34 and 69% of patients, depending on whether you use the most sort of conservative criteria for calling a mutation targetable. And we find driver alterations in almost all patients, but the most conservative definition, if there is a drug that's available, either approved or through clinical trials, where there's compelling clinical evidence that a patient with that marker will respond to that or could respond to that treatment, that would be our usual criteria. But there may also be preclinical or biological evidence as well. So there's a lot of interesting information that we find, but not all patients have a targetable alteration, and it varies pretty considerably across cancer types. So that's really allowed us to perform further research, clinical research, to credential new targets and develop new drugs. So this MSK Impact program has been the basis for screening and enrolling patients into clinical trials and providing comprehensive data to allow correlative studies when the trials are complete to better characterize biomarkers of response. So just to sort of walk you through the evolution of clinical trial design, and some of you I'm sure are intimately familiar with this, When I first started MSK, the trials, well, a lot of trials did no screening at all. There'd be a trial for patients with lung cancer where they'd be enrolled and treated with a particular drug, and maybe a separate trial for other patients, or even the same patients with lung cancer with a different drug. So with the emergence of targeted therapies, where there was a hypothesis that patients with a particular mutation may respond to those drugs, that led to the introduction of screening for which patients carried that relevant biomarker. So typically the screening was performed using an assay within the context of the clinical trial. So you had one set of patients that were enrolled into the study. They were tested first with a specific test for a specific marker, and the subset of patients that were positive, it could be a very small subset, would be treated with the drug. And a large number of patients could be enrolled into a separate trial, screened with a separate test, and then the tiny subset would be treated on that. But at our center, we've used the MSK Impact program as sort of a single engine for screening patients for all possible markers that might qualify them for different studies. So there's one single screening pipeline decoupled from the therapeutic study that allows us to identify patients with all the different mutations that might qualify them for all the different trials that would be open. So this has been really valuable in a specific type of clinical trial that was, I think fair to say, pioneered at MSK, called the basket trial, where rather than having all comers with lung cancer or all comers with breast cancer, patients with any type of cancer could be eligible based on the presence of a specific genomic biomarker. So only patients that were positive for that mutation could be enrolled, but any patient across any type of cancer could be eligible. And these basket trials have proliferated and led to the approvals, including some recent pan-cancer approvals of a number of targeted therapies. But it's tricky. It's tricky even for our oncologists working within their disease area to know what trials might be available, what the eligibility criteria are. So because of the availability of our data in a database, Tom said we don't want PDF reports, we want data in a database, we can integrate those MSK Impact results with all kinds of other diagnostic data, clinical data, even scheduling data. So all of this from our institutional database can trigger automated email alerts informing an oncologist or provider of actually informs an oncologist of clinical trials that may be available to their patient, and the email could be timed with their next visit or their next scan, and also inform the investigators leading clinical trials of a new patient that has a mutation that might make them eligible. So that allows those connections to be made. And because of the size of the sequencing program and the integrated data, MSK has been able to take the lead on a number of these basket trials, some of which have led to drug approvals. So each of these here are 3 different basket trials where MSK was one of 20 or more different centers, but because of the ability to identify these patients, we were able to enroll the majority of patients on each of these studies, and our oncologists are able to take sort of academic leadership in these trials. So it's been really important for the clinical research program. And then as a whole, as I mentioned, over 88,000 tumors sequenced, this is a huge amount of data that can be mined for the characterization of better biomarkers. Each of these genomic records is linked to a patient that's had some interaction with our clinical program. It could have just been a second opinion, or maybe they were followed longitudinally throughout their care. So we, across a number of our research labs, have formed a pan-cancer working group to mine and leverage this huge data set. Chai Banlamudi is a faculty member in my group who's taken charge and has shown that the resolution of this information has increased pretty considerably, even just in the last few years. So we had published about 5 years ago now our experience having sequenced the first 10,000 patients with MSK Impact, and could, with reasonably high resolution, identify the frequency of mutations in different genes across different types of cancer. But now this has sort of taken on a new form. We can drill down to individual cancer subtypes. We can look at a lot more genes. We can look at individual mutations and better understand which mutations are drivers and which ones are passengers. Oh, I lied. I was talking to Tom. I said I didn't think I had a screenshot of the SeaBio Portal, but I don't think I can give a talk without showing the SeaBio Portal. This is data from the AACR-Genie Consortium that we've contributed to. Other cancer centers around the country and around the world have contributed to. Vanderbilt locally is one of the main contributors, where all of our centers that are doing clinical cancer sequencing have aggregated our results into the SeaBio Portal. It's developed locally at MSK and is used as the means for sharing these data. And in the latest AACR Genie release, there are over 150,000 tumors that are sequenced. If you look by center, MSK, because of the size of our program, we're the blue wedge, so we're actually very sort of committed to data sharing and making sure that these results are available to the broader clinical and scientific community. So that's sort of the scope of our program, but we're trying to maximize the information that we get. And it's not just about making a list of mutations and hoping that one or more of those may be linked to a treatment. So what else can we learn from these gene panels? That's the basis for a lot of the research in my lab. And for this, we really are able to leverage the fact that we're doing this matched tumor normal sequencing. There's a lot of data. It's a paired analysis where in the tumor DNA, we can identify the somatic mutations, whereas all cells should have the same germline variants. And then there's a set of acquired mutations in the blood that's absent from the tumor that represents clonal hematopoiesis, or CHIP. So for the somatic mutations, I won't say too much more about this, but the ability to use the normal to filter out germline variants and make a comprehensive list of what's truly somatic allows us to be more precise about the individual alterations that are reported. And that's the left column. And this I already mentioned. These are the different classes that appear in our clinical MSK Impact reports. But there's this whole expanded set of what's currently a research result. But by looking at the prevalence of some of these more complex genomic features across our cohort, we can identify which of these might have clinical value. So we can we can identify a broader range of mutational signatures and mutation patterns. Things like whole genome doubling. When tumors have defects in cell division and mitosis, they often acquire extra copies of the genome followed by loss of individual chromosomes. This can be recognized as a poor prognostic factor. The mutation itself, I'll hold that thought for the next few slides. There are other features like HLA type, neoantigen prediction, viral DNA that we know we can identify from these data sets. So, an important feature is not just for the given mutations that we report, it's typically just listed as present. But more than just the presence or absence of individual mutations, we can ask whether a mutation is somatic or germline, whether it's clonal or subclonal, present in all the tumor cells or just a subset, whether it's heterozygous in one of the two copies or homozygous, or if through gains and losses of chromosomes, if the mutant copy and the wild type copy are imbalanced through extra gains of the mutation. And these turn out to be important features. They're not routinely reported clinically, but through the research that we're doing, I think we're moving in that direction. So for that last point, there are, this cartoon shows, there are lots of ways of introducing what we call allelic imbalance. If you have two alleles, a mutant copy and a wild type copy, through gains and losses, you can enrich one versus the other. And what we found, and this is a paper that was published a few years ago, was that for patients with pancreatic cancer or patients with colorectal cancer that have the same KRAS mutations, if you split the patients into whether those mutations are balanced, a single copy of the KRAS, and that's shown in the blue curves, versus imbalanced, where there's an enrichment of the mutant copy with respect to the wild type copy, usually through a gain of the mutation. All of these patients have the same KRAS mutations reported in their reports, but if there's imbalance, you can see that the outcomes are significantly worse. So this is something that may have prognostic value and the kind of feature that we're looking to report in the future. Also, the matched normal allows us to be more accurate with the tumor mutation burden that we report, because we can appropriately filter out all of the germline variants, which, if not handled properly, can confound those estimates. And this is actually from a paper published last month from Dana-Farber, who does their own clinical sequencing using tumor and not normal, where they looked across the different AACR Genie sites and showed that the centers that use matched normal, MSK on the left and Yale on the right, are able to most effectively filter out the germline variants, which is sort of that excess of mutations around the 50% variant allele frequency that you can see around the dotted line. And very importantly, because when you don't use a matched normal, that filtering typically involves population databases, that underrepresented populations, Asian populations, Black populations, are less effective in using these population databases to filter out germline variants and give more of a skew in the TMB that's estimated. So there are a lot of benefits, as I think already was alluded to in some of the questions, for sequencing matched normal. But the matched normal gives a lot of other information as well. So it allows us to identify germline variants definitively that may be pathogenic and lead to increased cancer risk, or in some cases may represent a therapeutic target themselves. So I won't say much about this, but having now done this germline analysis and reported back germline results in over 25,000 patients with the MSK Impact data, 17% have what's classified as a pathogenic or likely pathogenic germline variant. That means 83% don't, or at least not one that we recognize. But for these 17%, even if that cancer isn't dependent on that germline finding, there are implications for family screening and follow-up counseling and cascade testing in families. And then for clonal hematopoiesis, again, these are acquired mutations in hematopoietic or blood cells that lead to a clonal expansion. And through research that's been published over the last 5, 8, 7 years, it's now recognized that the presence of those clonal hematopoiesis mutations, while common and strongly associated with age, the older you are, the more likely you are to have a clonal hematopoiesis mutation that's detectable. These are risk factors for hematologic cancers as well as cardiovascular disease. And we've now been able to detect clonal hematopoiesis mutations in 25 to 30% of patients with solid tumors that we sequence with MSK Impact. It's an older population compared to normal, and also we're sequencing to deep coverage, which allows us high sensitivity for detecting these. But we've seen in our data there's a significantly increased risk of hematologic cancers for this subset. This was the rationale for MSK to establish the first ever clonal hematopoiesis clinic for patients with incidental findings of clonal hematopoiesis, otherwise favorable prognosis for their cancer, but are at greater risk of developing secondary hematologic malignancy, and because of that risk are routinely surveilled and screened for leukemia and other malignancies. So moving forward, what are we looking to do? I mean, we want to build on this research. We want to expand our reports, but also look at additional populations. So the focus up until now has primarily been patients with metastatic cancer. Initially, these were patients that were eligible for clinical trials and also most likely to benefit from this knowledge based on the available targeted and then immunotherapies. But there's prognostic value, as we're learning, for early stage cancers. There are rare cancer subtypes that we don't have enough information to recognize all the drivers, and underrepresented, underserved populations we think have the potential to benefit to the same degree as any person with cancer. But we recognize that within our population, within our clinical cohort on the left, the demographics are skewed. They don't reflect the U.S. population. They don't reflect certainly the New York City population, and efforts through partnerships with community hospitals and health systems are hopefully going to achieve better equity and balance. Novel diagnostic tests. So I intentionally focused on the tumor sequencing. There were already some talk about liquid biopsy approaches. We're very excited about the potential for plasma cell-free DNA sequencing. It has many advantages over tissue that I think most of you recognize. There's an expanded set of clinical applications. So it's not just about listing the individual mutations and performing comprehensive genomic profiling. There are questions about the levels of tumor-derived DNA and how that might relate to disease burden, or whether a patient's responding to treatment, or whether we can detect ctDNA at all, which can be useful for detecting minimal residual disease after surgery or earlier detection, which is going to be, I'm sure, the topic of the next talk. To address this, we've established sort of a companion test called MSK Access that was designed off of MSK Impact, capturing the most informative regions of MSK Impact, but for these sort of the smaller set of targets to sequencing to higher coverage, and we've now run this approved clinical test for over 7,000 cell-free DNA samples. And then finally, we want to broaden beyond just this list of targets. As Tom said, there are a number of patients where we can identify a clear driver, or at least a targetable driver. And by moving towards whole genome and whole transcriptome sequencing, We hope we can more comprehensively identify the full set of alterations, including structural rearrangements and signatures like homologous recombination deficiency, and then particular gene expression patterns, the infiltration of immune cells and immune-related patterns as well. So all of this, we hope, can serve as an integrated, as an opportunity to integrate across different platforms and imaging and enable modeling and provide a more complete picture and comprehensive diagnostic recommendation, a diagnostic description and treatment recommendation. So I'll end there acknowledging the many people in our molecular diagnostic service that are sort of the engine for keeping the MSK Impact and MSK Access programs going, and also members of my lab that are involved in the research with others that I presented here. So thanks very much.
Awesome. Thank you. So we're all going to take 2 questions. The first one's from Dr. Viana at West Clinic. How do you remind physicians at Memorial Sloan Kettering about molecular alterations, for instance, that may be on a trial that has yet to open, you surface it later on, or perhaps, as we're always concerned about, a new FDA approval and testing may have done previously. So do you have a way to implement that as an automatic reminder system?
In part, and we benefit from being one hospital. So if a new trial opens, we can generate, and this is routinely done for any new trial, particularly involving a targeted therapy, the PI of that study can get a list of all the patients in our database that might meet the criteria and create these automatic queries so they're notified, even if the patient's already come through and they were tested a while back and their oncologist isn't sort of thinking about the molecular report at front of mind. But I think one of the challenges is that when we issue a report, the information, the clinical annotations from OncoKB are static and they're current as of that moment in time. But we use the SeaBio Portal as an internal tool for updating those annotations as well. So the evolution of this is sort of an interesting story in its own. But the portal, which was developed as a research tool, is now linked directly from within a patient's medical record. It's a one-way link. It's available in de-identified form for everybody at our center. But for the treating team, they can go from the patient's record to that corresponding page in the portal where the OncoKB annotations are updated in real time. So that makes sure that at least the oncologist has that information. But we don't alert. We don't have alerts set up for a change in status or OncoKB, FDA approval, or the drug went from level 3 to level 1. There are communications that go out, but I think that's still an area where we can improve.
I think it's a huge gap for everyone. I think we're trying to figure that out, but it's very manual and hard. Second one is moderator's prerogative. I'm going to do this one. Yeah. So it feels like it's a Wild West right now on molecular testing at times. Not so much the somatic testing, but these newer areas you were discussing. Disease monitoring, early detection, MRD, and predictive models. We all understand the tipping point when it's a new drug approval, to be quite honest, but not so much in the testing realm, partly because Medicare payment is important, but there are ways around that. Is there hope that there will be more added clarity?
I hope so. I think it is confusing. I think with cell-free DNA in particular, there are a lot of factors. There's a lot of companies, it's a lot of hype, but also there's a wide variety of clinical applications. It's not just, as you said, about providing a comprehensive genomic profile. There are all these other things, MRD, early detection, and there's no one test that's good for everything. So there are niches that different companies are pursuing, but it makes the space more confusing and more crowded, and I think that's something that we're all grappling with. Within MSK, we're grappling with that as well. We have our MSK Access test that was designed to address the most immediate clinical needs, but it's not going to be as sensitive as other tests for early detection or for MRD. And we have a number of people, and the companies are going straight to the oncologist, and it's easy for them to collect blood in their clinic, and they're sending them out, and we're still trying to get a handle on everything that's being done and coordinate activities so we can learn from our experience collectively across these different partnerships.
Awesome. Thank you.
Yeah,
Dr.
thank
Berger,
you.
thank you for your time.
Sure.